Hello everyone, I'm Youfang. I'm from Department of Physics, a third-year PhD candidate. I'm glad to be here to present my recent research. This research is mainly about building a model to predict cell survivals. Predicting cell survival accurately is actually important in designing a good treatment plan, but in proton therapy, it is challenging to develop such model with good generality. Because in previous studies, cell survival is typically not just a function of dose, but also on other quantities. On the other hand, DNA damage actually is a fundamental variable in determining cell survivals. But the exact form between cell survivals and the DNA damage is not known to us. We then propose to construct a deep neural network to predict cell survivals from the DNA damages. To make this network biologically meaningful, we also trying to build that network according to major pathways of DNA repair. To make the training meaningful, we also need a lot of data, and it means many proton checks need to be simulated, requiring a lot of computing resources. Thus, we used our GPU-based G-micro-MC to do this work. This simulation actually cannot be done without the CPU-based packages. So figure 1 shows the overall ideas of our work. In the middle of this figure, it shows the network. So each node shows a subneural network representing the different DNA repair pathways by its name here. The input to this model is the DNA damage with different complexities, and its output is the prediction of cell survivals. This DNA damage spectrum is calculated from Monte Carlo simulation with different conditions determined by the proton experiment. In this way, these predicted cell survivals can be compared to the measured cell survivals. So the process or the goal of this neural network is trying to minimize the difference, square difference between these two cell survivals. We did simulation on 12 different conditions, and we trained the network on the middle range, the LET, while we tested on 2 LET that is outside of this range. And here on the right, it shows the result. So first, as you can see, the DNA damage spectrum is not uniform because for certain doses, there exists a wide range of DNA damages. This non-uniform DNA damage actually suggested the average dose actually is not a good variable to generalize model. So now let's see the results from neural network. So on figure A and B here, it shows the performance of this neural network on training data and testing data. As you can see here, it performs well on both data sets. And as comparison, another linear quadratic model where cell survival is just simple functions of dose. And by fitting the data in the middle range, we can get alpha beta distribution, and by using polynomial extensions, we can also get the alpha beta value outside the middle range. So here, figure D shows the predictions and the ground truth. As you can see here, this model actually performs worse than our neural network. The relative error is 0.1 versus 0.23 for this one. So another important thing for this neural network is the interpretation. We then examined the network weight for this single-stranded break related pathway and this double-stranded break related pathways. We found the DSB related pathways is usually much larger than the single-stranded break one, and indicating the importance of the double-stranded break in cell kills on the proton radiation. So to summarize this work briefly, we used our in-house developed GPU-based Monte Carlo simulations to get the DNA damage spectrums, and then we built a network to predict cell survivals from this DNA damage spectrums. This model actually can have a unified universal parameters for different LETs, indicating its generality. Our future work will be focused on the robustness of this network and more depth interpretation of the model parameters. Thank you. That's all.